Hello, my name is Marcin Joachimiak, and today I'll tell you about understanding Earth's ecosystems using machine learning. I work at Berkeley Lab, as well as the DOE K-Base, and K-Base is a system that aims to empower scientists and teams to collaboratively perform computational biology on plants, microbes, and their communities. In the top right, I'm showing a series of core elements of the system. There's user and reference data with standardized types, open source tools wrapped as apps, and customizable shareable analysis notebooks to perform the analysis, including using these apps. On top of this, we're building something called the knowledge engine. This is an internal R&D project to support prediction and inference on the system. Before I dive in, I wanted to share some numbers from K-Base for the last year. We had continued user and reference data growth, as well as growth in the number of analysis narratives, and continued publication output. The reason this is important is because this is all based on standardized data types, and so provides a solid foundation for developing AI ML approaches such as the knowledge engine. So we set out to explore Earth's ecosystems using metagenomes. We know that our planet is made up of a network of interconnected biomes that can be categorized into types. However, our understanding is still limited. For example, we know little about the effects of processes like climate change and interactions between these ecosystems. We can consider metagenomes as snapshots of biome communities and return to the EBI Magnify resource, which provides data based on published, open, standardized pipelines, along with human curation. To our knowledge, this is the largest collection of assembled, annotated, standardized metagenome data out there. We're interested in learning associations between ecosystems and these metagenomic features. So in the top left, we start with experiments. Samples are physically generated from the field or from host anatomical sites, and these are processed into reads using sequencers. Next, a series of computational steps follow. The reads are assembled into contigs. The contigs are annotated with protein domains, functions, and taxa, in this case, presence of 16S on contigs. We have a number of underlying ontological namespaces that support this analysis. However, for environmental features, that data is poorly standardized and not amenable to this analysis. Importantly, the ecosystems are classified using the Gold environmental classification, and that was one aspect of the EBI curation. We expect to discover features that are adaptive for specific ecosystems. For example, presence of oxygen could be associated with aerobic metabolism and oxidative stress functions. Early on, we performed an analysis that we continued to hone, and this is comparing samples pairwise to the averages of ecosystems. What we see is that at times, the original assigned label is less similar than some other ecosystem. And so here on the example of aquatic marine samples, almost half of the samples are more similar to aquatic lentic brackish environments, suggesting a potential more detailed labeling here. And there are other examples, allotic sediment, hydrothermal vents, estuary, along with some oddities such as fermented beverages and large intestine fecal. So in this table, I'm listing the different cases of limits of labeling. There's classification degeneracy, there's potential for contamination, the samples can come from mixed environments, the label could be overly broad, as in this case of aquatic marine, or they could just be mislabeled. And about 20% of the samples in the data have this behavior. However, there's a very important caveat to note, and that is these similarities are computed using all features, and this can introduce noise and bias. Our machine learning task was to predict sample ecosystem labels. To do this, we turned to two different methods. First, we use gradient-boosted decision trees. This is a strong learner that's made up of a sequence of connected weak learners, where at each subsequent step, you're trying to fix errors from the previous step. This process ends up being highly efficient and leads to fairly accurate models, often rivaling deep learning methods. We also tried TabNet, a deep tabular learning method that uses a sequential attention mechanism to choose features to reason at each decision step. And this has some interpretability and efficiency gains. As you may know, these types of projects often have a long lead up involving data processing, pre-processing, and cleaning. So we start on the left with calls to the EBI Magnify API. We obtain Go term features, IPR domains, and taxa. We merge these together, and then through a series of computational steps, we prepare the data for training and other aspects of model evaluation. We require that an ecosystem has at least 15 samples, and we remove collinear features and retain a representative. And there are other things going on here, but to just summarize, we're left with 41 ecosystems, 19,000 features, and 18,000 samples. Using this data, we set out to train a series of models using individual and combined data types. So the table here is based on hyperparam optimized models, as well as tenfold cross-validation to report the mean test data accuracy. The top model was CatBoost with all 3 feature types, or with IPR domains alone, closely followed by TabNet with all feature types. However, this method took longer to train. We focus on the top full resolution model with all 3 feature types for ease of interpretability, and you'll see why later. On the right is another series of models. These are lower resolution models that are trained on roll-ups of this environmental classification. At each roll-ups level, we have fewer and fewer classes to predict, and so the problem becomes easier, and these models, as you expect, perform better, of course, with the loss of resolution. Now we're interested in the model performance, and the confusion matrix in the top right shows along the diagonal in dark blue all the ecosystems that were perfectly or near perfectly predicted. However, the model, of course, performed errors as well, and we're actually able to classify these errors using 4 different semantic relations. Nearly all the errors were accounted for with this set. So first, we have possible contamination or mixture. For example, food production samples predicted as large intestine fecal, somewhat worryingly. Next, we have hyperonyms and hyponyms. These are terms that are lower or higher in the classification. So not exactly wrong, but just not at the right level, still potentially useful. And finally, we have contextual synonyms. These have to do with the degeneracy in the classification. For example, wastewater could be encoded as engineered or host associated. I mentioned earlier that we required a minimum number of samplers for training, and this left about 300 samples from rare ecosystems that the model had never seen. So we thought this was an opportunity to make an assessment of the model generalizability. In green is the line from random expectation, where you're randomly predicting a possible label from at that level of the classification. And this has a sharp drop-off as the resolution increases and the number of labels explodes. In red is the line based on semantic similarity that measures the overlap in the individual terms in the classification path between the predicted label and the true label from the unseen ecosystems. And you can see that the model does much better than random, even at the higher resolution levels. So we think there is some degree of generalizability from this analysis. Now, we were interested in the model performance, but I think we're much more interested in what the models had learned and the features and their biological interpretation. So to do this, we created N data splits, each with their own copies of permuted features, and we trained N models. Using those results, we're able to compute an average feature rank using the positions of the permuted features as natural thresholds. And the bottom is a heat map of the input data for this 568 important feature set. And the data clustering shows very good agreement with the top 3 level categories, as well as finer structure in this classification. And importantly, the hierarchy of that classification was never used in training. In addition to the feature importance set, we also have the feature importance values themselves. So positive importance means that the feature is contributing information to the class prediction, and negative importance means that it contributes confusion for that class. On the bottom is a heat map of the binarized importance values. And you can see labeled a few different classes of behavior of these features. They can be rarely important for a small number of ecosystems, mostly positive, mostly negative, and of course mixed. We also trained 2 additional models using the feature importance sets, either at the ecosystem-specific level or for the overall model, and these had comparable or slightly better performance, suggesting that potential further feature selection could lead to small gains. We developed some vignettes for some important features to highlight their biology. So the top 2 here were extremely important for some human ecosystems. Both of them are transporters, and they actually have flipped importance meaning between skin and some other and digestive ecosystems, for example. And on the bottom, we have 2 features that are interesting biologically that have kind of weaker or maybe questionable importance. But we're particularly struck by this visible detection of light feature, which was important for marine ecosystems, as you would expect, but also showed some weak importance for large intestine and fecal. Now, you can ask, why are phytochromes present in gut microbes? But first, I have to tell you that the importance for that feature was actually due to the absence of that feature. However, we're still able to find some examples of phytochromes in the human digestive ecosystems. So overall, we had thousands of examples of bacterial and other phytochromes in the overall data set, and just a handful of examples in this digestive ecosystem, including one from corn. And so we think these are kind of secondary biomarkers that are due to diet or environment that are traveling with that sample, so representing more a mixed case or maybe even contamination, if you wish. However, the phytochrome story itself is quite interesting. So there are known examples of bacterial phytochromes that can utilize a mammalian tissue chromophore, and these are in fact the most near infrared shifted towards the tissue transparency window. In addition, there's some recent results that show that certain phytochromes can detect heat. However, even though we have this kind of very exciting, interesting findings in our hands in this data, the explanation was more mundane. And I think this highlights some complexities of metagenome data and also the difficulties in validating and going through all the layers to convince yourself. To further validate the important feature set, we performed some tanglegram clustering tree comparisons. So on the left is the tree based on all features. On the right, and next to it is important and trained similar features. Then trained similar features were ones that were removed during the collinear filter, and now they're recruited back here for completeness and to aid the interpretation. So you can see that these two trees are nearly identical. They have some just very small inconsequential changes of the leaves. Now on the right, we're comparing the important and untrained similar data clustering to the gold classification hierarchy itself. In this case, the entanglement is more complex. However, you'll notice that the top three level categories have very good agreement. And moreover, some of these big movers, the solid lines, suggest some degeneracy in the classification. For example, coral reef could be encoded as environmental or host associated. So you think based on these results that the important features are also representing some higher order relationships in this ecosystem classification. So finally, we wanted to understand how these similar features, important features relate. And this is the breakdown of that feature set. You'll notice that about 20% of these are domains of unknown function. That is now an active side project as part of the knowledge engine work. But this is what the similarity network looks like. And I mentioned early on the choice of the IPR domain only model versus the mixed data type model. So unfortunately, interpro domains have nice coverage and performance, but they're less interpretable for two reasons. Some of them don't have a really assigned function, but many of them link to full-length proteins that have additional features that impact their function. So that domain is not a really good stand-in for the protein level information. So it's very nice to have a sprinkling here of Go terms and taxa, which are more interpretable and can aid in interpreting the neighborhoods here in this network, including for some of those interpro domains. And that work is ongoing. We also computed a network for the ecosystems based on similarity relationships induced by the important features. The node size here represents between centrality, so a measure of node influence on the network. And you can see very nice recapitulation of the top three level categories: environmental, engineered, host associated. However, there's some very interesting outliers: host associated plants rhizosphere, phyloplane, human skin, and human skin neris. Now you may recognize that all of these ecosystems, even though they're host associated, are host surfaces that are exposed to the external environment. And so in fact, it makes sense that they exhibit similarity, or maybe even larger similarity, to other types of ecosystems than to host ones. So we think this suggests that the ecosystem relationships are better represented by a network than by a tree. So to conclude, machine learning can reduce feature noise and improve interpretability. About 20% of all features can retain ecosystem relationships and much of the environmental hierarchy. Now we're using these important features as a data product and resource in multiple projects, including to support biological discovery. And finally, we need more of certain things: high quality data, metadata, curation, standardization, and unfortunately, memory. So finally, I'd like to acknowledge the overall K-Base team, and special thanks to the K-Base KE team: Zeming, Sean, Bill, Prachi, Chris, Megan, Shane, Paramveer, and Adam. Thank you.